So mutations and evolution. If the mutation, now we've talked about this before, but I just want to reiterate, mutations are random. Now, if the mutation damages the organism's going to have poor survival rate, not going to pass on that mutation, or less likely to at least. If the mutation has a neutral effect, like a silent mutation, for example, it's going to have no effect on survival. Maybe it'll get passed on, maybe it won't. It's up to other factors, right? It's up to other chance factors whether that organism is going to survive, pass on its DNA to the next generation or not. Okay, so no effect on survival. If the mutation benefits the organism, then the organism is going to have an improved survival rate, better chances of having more offspring and passing on its genes to the next generation. Then the mutation is going to get passed on generation after generation after generation. Okay. So mutations affect unless they're neutral, mutations usually affect the survival rate of the cell that experiences the mutation, and also will affect any progeny it's able to have, if any, because the mutation is heritable. We inherit DNA from our parents. Improved survival rate over competitors means faster rate of growth, fast and spread of the new mutation, and that creates genetic diversity, because what a mutation does is it creates a new version of a protein, new versions of a gene. So we have diversity because of mutations. All people used to have brown skin, right? There's this enzyme there's a series of enzymes that produce a pigment called melanin. Melanin is brown and it's a natural sunscreen. Those of us who have white skin, like me, I'm super pale, I burn super easy, I've got freckles. So I have inherited from my ancestors a mutation to an enzyme responsible for producing melanin. It's a dysfunctional enzyme. It doesn't make that melanin color anymore. I have no natural sunscreen and I'm living in this desert and it's bad for my skin, right? So that is an example. So and we talked about with the penicillin, the penicillin resistance, we talked about how there's a trade-off usually. So a mutation might be good in one environment and bad in another. So if you look at the distribution of different skin tones around the globe, you'll notice that in places with really intense sun, you usually see browner skin. In places that are cloudy, like Scotland, and always raining and you never see the sunlight, that's usually where you see paler skin. Now, if you don't see the sun, you don't need the natural sunscreen, and also vitamin D is really hard to come by because we can only produce vitamin D in the presence of sunlight. And so dark skin gives a natural sunscreen, but it also makes it harder to produce vitamin D. Now, if you're experiencing sun onslaught all the time, you don't have to worry about making enough vitamin D because there's plenty of sunlight anyway, even through that sunscreen. But if you live where you never see the sun, it's going to be really hard to make vitamin D, and it's actually a little bit better to have lighter skin in that case because it makes it easier to use the little bit of sunlight that there is and still make enough vitamin D. Okay. So different mutations are good in different environments. It's the same thing with the penicillin resistance. The transpeptidase enzyme that builds the cell wall, when it's mutated, doesn't build the cell wall as well. So that's not good under most conditions, but it just so happens that same mutations makes it impossible for penicillin to gunk up the enzyme. So that's really good if the bacteria ever encounters an antibiotic. So there's a trade-off, and whether the trade-off is worth it or not just depends on what environment you find yourself. All right, so here's just this chart again, just to drive this point home. A mutation that confers antibiotic resistance is only beneficial to a population of bacteria if it's exposed to antibiotics. So here you have a slightly diverse population. You have some resistant bacterial individuals, some resistant mutants, not very many, because this mutation isn't beneficial at all in natural circumstances. But you add the antibiotic, so these bacteria, they're not building cell walls very well. So they're kind of dying off. These mutants are not, they're struggling. The mutants are struggling. But then you add the antibiotic, these mutants, they're not building a cell wall as well, but they're the only ones that can survive the antibiotic. So now you've changed who's most fit for the local environment because you've changed the local environment by adding antibiotics. So under normal circumstances, these mutants, they're struggling. Their transpeptidase enzyme is a little bit dysfunctional. It's still working okay, but it's not working that well. So they're struggling. Add the antibiotics. Now these mutants with the mutant transpeptidase, they're the only ones to survive. All their competitors are cleared from the environment and they take over. Okay. Now this survival of this species can only happen because of the initial diversity. The mutation is not happening after the antibiotics. So again, they're not willing themselves to become resistant. They don't have that capability. They can't just snap their fingers, their little microbial fingers, and say, oh, there's an antibiotic here, let's become antibiotic resistant. No, they don't have that power. We don't have that power. All that happens is the ones that are already there are going to survive or die depending on their traits. So this is why diversity is important to every species. Every species, diversity provides stability. Because if you have many different traits in the environment, then it's more likely that when the environment changes, when stresses change, there's going to be somebody around who has the traits to survive the environment. If everybody has different strengths, different weaknesses, you can't predict the future. You can't predict how things are going to change. So if everybody is different, everybody has different strengths, everybody has different weaknesses, then when the environment changes, different individuals are going to die off, different individuals are going to survive. There's more likelihood that somebody will survive the change. Versus if everybody is a true clone, if everybody is exactly the same, let's just say there was no diversity in this population. Let's just say all the bacteria were true clones of each other, none of them were resistant mutants. Well, then the bacteria would just die off, right? And so when the Earth changes all the time, environments change. There are hurricanes, volcanoes, fires, species invade, species disappear. There's global cooling and heating cycles. We have ice ages that alternate with tropical ages, right? So the environment's always changing. Species that don't have a trait to allow them to survive the change go extinct. Species where some individuals do have traits that allow them to survive the change evolve. So here is a diagram of evolution. If there was no diversity, if there were no resistant mutants, you would see extinction. So we usually think of bacteria as a clonal species. They do replicate by clonal reproduction, but when they're not replicating, they are able to intentionally increase their diversity by trading genes with other species. And that's what we're going to talk about now. How do bacteria get diversity? If diversity is so important to survival, sexual reproduction, that's great for diversity because every offspring is different. Every offspring is unique. So we've got all kinds of diversity in the human population, mammals, plants. Sexual reproduction just makes lots of diversity. But what about clonal species like bacteria? If diversity is so important to survival, how are they surviving with clonal reproduction? The answer is they have, there's, yes, mutations, but mutations are still kind of rare. Bacteria and archaea, they have evolved a whole other means that's different from sexual reproduction just for increasing genetic diversity. And that's what we'll talk about next.